Hello everyone. Today we are going to talk about microscope. What is microscope? An ecosystem consists of different variety of organisms. Some of them are unicellular, means made of one cell. Others are multicellular, means made of more than one cell. They all need to survive. Their vital activities allowing them to survive take place within the cells. The question is, do you think you can see the cell by your naked eyes? The cells are too small to be seen by naked eyes. So to overcome this problem, some scientists have developed the magnifying instruments such as hand lens and microscope. But before, what is a cell? A cell is a structural and functional unit of organism. Yes, I think you got it. A cell is a structural and functional unit of organism. So to see the cells, we have to use the magnifying instrument because the cells are too small. Except an egg cell, only one cell seen by the naked eyes is egg cell. So generally, what is microscopy? Microscopy is a technical use of microscope. So what is microscope? Microscope is a device used to produce a magnified image of an object or specimen. Microscope was introduced at the first time by Anton van Leeuwenhoek. The microscope used was enough to explore the microbes. The most common used today are grouped in two types. One, compound light microscope. Two, electron microscope. One, compound light microscope is also known as optic microscope. How does it work? The light passes through a specimen, then through two sets of glass lens, the objective lens and the ocular or eyepiece lens. I think you got it. I repeat once again. Light passes through a specimen and then through two sets of glass lens, the objective lens and the ocular or eyepiece lens. The lens refracts or bends the light to give a magnified image of an object. The image may be projected directly into the viewer's eyes or onto a photographic films. The image may be projected directly into the viewer's eyes or onto a photographic film. The question is, what is a photograph? A photograph is taken through a light microscope. That photograph taken through a light microscope called photomicrograph or light micrograph is called photomicrograph or light micrograph. Usually, light compound microscope consists of four types of lens or objective lens. Times 4, times 10, times 40, times 100. Magnification and resolution. magnification and resolution. In dark, I repeat, in dark, a car's headlight at some distance away appear as one light source. I repeat, a car's headlight at some distance away appears as one light source. If taking photograph, it will also show one light source. Now, what is magnification? Magnification means increase in apparent size of object or increase the size and shape of object. Why do we increase the size of an object? The object size is increased to be seen clearly. No limit to the magnification produced by a light microscope. I repeat, no limit of the magnification produced by a light microscope. This means it depends on the power of the lens used. I repeat, it depends on the power of the lens used. Second, above a certain magnification, the image becomes unclear. I repeat, above a certain magnification, the image becomes unclear. At this point, it is impossible to distinguish structures laying together. At this point, it is impossible to distinguish structures laying together. So, what is resolution? Resolution is ability to see two distinct or separate points or objects. Ability to see two distinct or separate points of object. Notice, the resolving power of light microscope is limited by the wavelength of light. Yes, you get it. The resolving power of light microscope is limited by the wavelength of light. The light microscope can magnify object up to about 1,500 times without losing clarity. The light microscope can magnify objects up to about 1,500 times without losing clarity. Now, if we have understood the difference between magnification and resolution, now we can see how to calculate magnification. How to calculate magnification. Magnification equals to size of image over size of object. I repeat, size of image over size of object. An example, if a nucleus has 100 millimeter on a diagram with magnification of times 10,000, what is the actual size of the nucleus? Repeat the question. If a nucleus has 100 millimeter on a diagram with the magnification of times 10,000, what is the actual size of nucleus? Actual size equals to image size over magnification. Actual size equals to image size over magnification. This means 100 millimeter over 10,000 equals 0.01 millimeter. Thank you for hearing this information. See you next time.